And next talk is by Dr. Ratnam Rai. He will present on the quantitative analysis of illumination and detection correction in adaptive light sheet fluorescence microscopy. Let's hear the speaker introduction from Andrew.
So, Dr. Mani Ray is a postdoctoral researcher in the Greenbaum lab at the Joint Department of Biomedical Engineering at the University of North Carolina and North Carolina State University, United States of America. He received his Ph.D. degree in electrical and computer engineering at Ben-Gurion University of Negev, Israel. He has authored or co-authored more than 30 international journal articles and has presented several papers at international conferences. His research interests are in the field of digital holography, computational optics, microscopy, biomedical imaging, light sheet microscopy, deep learning, etc.
Thank you for the kind introduction. Can you guys see my slides and can you guys hear me nicely?
Yes, Mani, you can.
Yeah, thank you. Thank you to everyone from the organizing committee for allowing me to present this talk. So the research that I'm presenting is quantitative analysis of illumination and detection correction in adaptive light sheet fluorescence microscopy. So I'll start with the motivation for this work. So I'll come into the motivation for this work. So currently around 1.5 million people suffer from hearing loss. And it has been estimated that 80% of them can be treated. So currently the drugs that are being developed, they are being tested in vitro, and they use basically classical histology techniques to image them. However, the problem.
Sorry for the interruption, Mani.
Yeah.
Can you try to change the slide? Because we are still seeing your first slide, like the title slide.
Oh, I am changing.
Yeah, but it's not showing us.
Yeah, now it changed, I think. It's motivation.
Yeah, now it changed.
Is it changing again? Can you see all the slides?
Yeah, it changes.
Yeah, it's working now. Yeah.
Okay, thank you. So, yeah, so currently they mostly use histological techniques to image the sample. However, the problem with histology techniques are they are two-dimensional techniques, which means that you take a thin slice of a sample, you image it. But when you try to image a full sample, it takes a lot of time to image the sample if you want to do the full 3D imaging, as well as it requires a lot of pre-processing and post-processing of the images to create the 3D map of it. And it has been avoided to do in other organs, but when it comes to the cochlea is a three-dimensional structure which has a spiral structure in it, and each point in the spiral structure actually corresponds to different frequency range that we can hear. So if you want to study it and want to know, okay, which frequency range we are having the problem, we need to have the 3D imaging needed, and classical histology technique cannot do that. However, there has been a few decades ago, a chemical technique called tissue clearing has been developed. What it does is basically it takes an organ and make it optically transparent. So now that we have an organ which is basically optically transparent, we don't need to slice it, because now the sample is transparent and we can do the entire 3D imaging of it. And most of the biological researchers use confocal microscopy for imaging these things. However, the problem with the confocal is, first, it's a slow imaging technique, which means that because being a point-by-point scanning method, and when you try to image the whole organ, which can be up to in size of up to multiples of mm's, it takes approximately, for this particular sample, as you can see, it takes up to 1 week to do the imaging. And when you're doing a imaging for a 1 week, and you're doing a fluorescence imaging, many various challenges happens. First, because the confocal is a epi-illumination type of microscopy system, it bleaches your sample, it bleaches your sample. In addition to imaging it for a week, it basically destroys your sample's fluorescence signal in first day of imaging. So this day of the imaging that you're trying to do the imaging, it actually gives you no signal or no SNR of the images result. However, light sheet microscopy, which has developed recently, few years back, it's a new type of 3D imaging technique which can do the imaging of the samples 100 times faster than the commercial confocal microscopy have, while maintaining the minimum photo bleaching and phototoxicity. So our plan was to image the cochlea using the light sheet microscopy. However, like when coming to the light sheet microscopy, so how does the light sheet microscopy work? So in light sheet microscopy, basically you generate a thin sheet of light, which is basically 1 to 10 micron in size, and you illuminate the sample through it. And from an orthogonal direction, you actually capture the fluorescence signal of it. So how it becomes better from the confocal is because you're not illuminating the whole sample, you're just illuminating a thin sheet of light. It doesn't photo bleaches your sample. Second is instead of point scanning method, it in one field of view, you can capture the whole image. So that actually helps. Is it still slide 2? Hopefully this is good. Is it changing?
Yeah, now it's full screen. Yeah, it's changing.
Yeah, okay. So I was at light sheet microscopy. So in light sheet microscopy, you basically use a sheet of light to illuminate the sample. And when you illuminate the sample with a sheet of light, it doesn't photo bleaches your sample. And other than the confocal where you have to do point by point scanning, here you capture the sheet that is used to illuminate the sample, and you can have the full field of view imaging. So it makes the imaging faster, and you can have the full entire 3D imaging of the samples without any pre-processing or post-processing involved or photo bleaching the sample. And these are the few examples of the imaging that we have done. This is an example of a mouse lung that we are able to image. And these are some of the particles that were present into that. So we were able to image that. And these are the bone structure of the rats with the cancer cell present over here. So these are the few examples that you can do the entire imaging of these samples in a continuous manner without any slicing or any type of human intervention. However, like any microscopy, light sheet also comes with its own problems. First is the defocus. The defocus is basically why it emerges in this particular case is because you are imaging a very large structure. So when you go within deep inside the tissue, the focal distance actually changes because of the change of the optical path. So the defocus happens over there. The second is the key problem in the light sheet is that the illumination light sheet that you are generating and the detection objective focal plane, they need to be coplanar. If they are not planar, then all the part which is outside the depth of field of your objective will not be get into the focus. So it actually results in a focus map, as you can see over here. Here, yeah. Over here, so you'll have a good quality image in the middle, whereas your imaging quality will start to deteriorate when you move, when your light sheet is not parallel to your focal plane. So that's on the illumination side. These are the two errors. Whereas on the detection side, the aberrations which are present in every imaging system is also present over here. Here, it becomes more complicated when you have a specimen, the sample-based aberrations. The sample-based, I mean, is that when you use tissue clearing method to make the tissue optically clear, it's not a perfect process. So there are some challenges associated with it. So if the quality of clearing is not good, it introduces aberrations. If you have a lot of transition between hard and soft tissue, which is basically you have a refractive index changes between when you move from one part of tissue to another part of tissues. So all these problems actually creates the aberration. And when you go more deep, they basically start to stack on one top of each other, and they becomes over there. Uh, in literature or in the research, people have corrected for these errors, and they have used adaptive optics for aberration corrections, or they have used pre-calibration method for it. However, the most of the research that don't talk about is the sample-based errors present or not. Most of the techniques, they discuss that, okay, we have once corrected our light sheet microscopy setup and it's all well, good and going. But the sample-based aberrations, people don't talk about it. And that's a question that we have answered in our research, is that what's the extent of sample-induced illumination and detection correction? The second, what should be the correct order? You have two type of errors in your system, what should be the order of correction, and what is the best way of correcting these errors? So these three questions I will be answering. So this is the optical system of adaptive light sheet that we have. So we have a laser and we have two galvo mirrors. So these galvo mirrors are basically used to generate the sheet of the light, and they can actually rotate the light sheet in any direction, whether it's in ZY or in ZX. And then you use an illumination objective to create the sheet of the light. The fluorescence signal is actually captured from a detection system and is modified from a deformer mirror for the adaptive optics. So first thing comes is quantification of errors. That how much is the extent of sample-based errors, illumination, and the detection wings. So for the illumination wings, we have the error of tilting of the light sheet. So there are two ways it can be done. The tilting can be in ZX direction or in the ZY directions in respect to detection lens. So we call it yaw and roll angles, and these are we get a tilt up to 6 degrees. To give into perspective, if we are doing a 10x magnification imaging, this tilt results in a deviation of your light sheet from a focal plane of up to 50 to 60 microns. And currently the depth of the field of the normal objective is somewhere between 20 to 30 microns. So which means that a tilt of 60 micron will have your 75% of the image will be out of the focus. Whereas when it comes to the detection side, so we estimated what are the major aberrations that are present in the samples, and we found that the comma astigmatism, trefoil, and the spherical, these are the 4 type of aberration, and these are the Zernike amplitudes that we have retrieved. That these all of them are being present when doing the imaging from the specimen. In all of these results, we have removed the baseline, which is basically the optical system. Whatever the irregularities of the optical systems are there, we have removed it. So all of these are just from the sample induced. So next comes correcting. So illumination correction is basically for the defocus, it's usually either done using focus stacking, whereas for the angle, it's usually done from the manual correction. And both of these is not good when you are trying to do for the live imaging techniques. So to solve this issue, we actually develop a deep learning based method for correcting these. So what we do is basically when we capture one image and we give a known defocus to it, we capture 2 images and we send it to our neural network, deep learning network that we have trained for it, and it actually calculates what's the roll and the yaw, basically what's the tilt of your light sheet is present. We correct for these corrections, and then just to see if there are any defocus present into the imaging system, we correct for the defocus also. And thus we have a pipeline which can estimate the amount of shift of the light sheet, whether it's in roll direction or in the yaw direction, as well as it also corrects for the defocus estimation. So instead of having 10 to 20 images to correct for all of this, we just use 2 images to correct for any illumination based error. Correction, and these are some of the results. So first image is basically you have the illumination errors, you have the roll. So the scale bar over here is basically how much defocus is present. So red means that you have minus 36 micron defocus from the focal plane, and on the other side you have the plus 36 micron. So these are the defocus. The amount of error is there, and after we do in the second line, when we do the angle correction, you can see the amount of defocus has become uniform. It means that yes, your image is not in focus, but the amount of defocus is same, which means that your light sheet has been corrected such that the plane of illumination and the detection both are parallel to each other. And in the third stage, we correct for the defocus, and as you can see over here in the third part of this images, that the amount of defocus present in the whole of the system is very minimalistic, and it's within the field of view of the imaging. So using this way, we were able to correct for the illumination error in light sheet microscopy. Next comes the detection. So in detection, we have the aberrations. So typically, people use wavefront sensor to correct for it. However, the problem in biological imaging using a wavefront sensor is that you are losing a lot of light. And the fluorescence imaging, if the amount of light you lose, the SNR of a system start to go down. The second, the most common use method is the iterative method. But the problem, again, similar to the previous cases, iterative method, you need multiple or many images, which basically photo breaches the sample. So here also, we developed a machine learning based system where you have 2 images and we feed feed into our neural network, and it actually estimates the different Zernike coefficient corresponding to different type of aberrations. So after we calculate, okay, these are the aberrations that we have present, we give it back to the our imaging system and correct for it. So this is just an example of the aberrated image and the corrected images that we have corrected using our deep network over here. So now that we have a smart adaptive light sheet microscopy in which it takes the images from the sensor, and it, using the deep neural network, it corrects for your illumination as well as it corrects for the detection also. However, now the next part comes. Okay, now we have correct for the both type of errors. What should be the strategic way of combining them? Is it just that we do one of them and it's more than enough, or we just simply run them, it will be okay? So to answer these questions, what we did is we did the, for the same field of view, we did different corrections. So we have an uncorrected image. You did only the detection correction. We did only the illumination correction. And then when we combined both of them, it can be done in two ways. We first do illumination and then detection, or vice versa. We captured the images after the correction, and we used DCT, which is basically a DC cosine transform energy, as a image quality matrix. And we saw that, okay, how much is the improvement of the imaging qualities present over there? So we saw that illumination correction has more share in the errors that is present in comparison to detection, and it can be answered in a way that because the illumination creates a 75% of your image which is not in focus, that's why it has more contribution. And when we are combining both of them, it is seen that the illumination followed by the detection correction has more effect in comparison to the detection followed by illumination. And it can be answered in a way that because in illumination correction you are shifting your light sheet, so by shifting the light sheet, it might be possible that you are illuminating a new field of view which has a new type of aberrations. Hence, correcting the illumination first will always be a better way of doing it. And these are the some of the results of this. These are the uncorrected and the corrected images. In uncorrected images, you have the aberration present all over the field of view. You have the defocus present on the both sides because of the tilt of the light sheet, whereas in case of the corrected image, full entire field of view is in focus, as the aberration has also been removed. So to conclude my talks, we have particularly specified that sample-based errors are present in the light sheet fluorescence microscopy, which has been ignored by many people. We have developed a smart light sheet microscopy, which uses a deep learning method and uses just few images to correct for whether it's a tilt of the light sheet, defocus, as well as the aberration in the system. And in future, currently our neural networks are working as a standalone system, which means that the illumination and detection doesn't works together. In future, our aim is to basically construct a final smart light sheet microscopy system in which you just have to place your sample, and it will automatically do the 3D imaging for you, whether it will just combine all the illumination and detection correction, and you'll have the best imaging quality. So finally, I would like to acknowledge Dr. Alon Greenbaum and all of my lab members at the NC State and UNC for conducting this research, and some of our collaborators, Professor Rob Turan, Professor S. Roy, and Professor Heather Pieperstone. Thank you.
Thank you, Dr. Mani Rai, for the excellent presentation. It is like interesting to know this talk like about calibrating the sample-based aberration. I have a couple of questions. So when you say about the sample-based aberration correction, for each sample loading, do you need to optimize the system, or
no?
How much time it would took?
So it's more of your optical system based rather than sample based. So suppose in the machine learning, the deep learning technique that we have developed, we have trained our model on a mouse brain. Okay, but the thing is, the input image that we send into the neural network is of 128 pixels. So it's like you're detecting the point object in the whole image. So any sample which has a point object like structures, we can use that to correct for it. So it doesn't need to be one type of specimen. It can be used for any type of specimens.
And also, in continuation of the question,
Yeah.
When you talk about the mouse brain or the neuron cell, it doesn't have the equal thickness, Z thickness. It has uneven Z thickness, right? So how do you differentiate? It is the tilt of the aberration or it is coming from the sample itself? Because when you train for the particular sample, that might be different with the other sample, right? The thickness. So how this model will work for that sample?
No, that's the thing. This is thickness independent. In light sheets, the thickness doesn't comes into the picture because you are just illuminating with a thin sheet of light. And when you go to the deep, and what we have seen in when you go deep into the sample where the light has to travel through whole specimen to reach your collection objective, the amount of aberration will be higher. That's the only difference that happens. At the surface, you will have very minimal aberration because you have corrected for your optical system aberrations. You have removed the optical system aberration. So it's only the specimen. So on the surface, it will be like 0 aberration is present. And when you go deep into the sample, you like 100 aberration is present. And we have calculated for all the numbers from 0 to 100, what will be the aberration will be there. So it's like that the whole system is developed. You are muted, sir.
Yeah, excellent. So I understand that point now. The question, the hand raised by Professor Vijay Kumar Anand. Yeah, now you can ask him.
Okay, thank you. So, Mani, thank you very much for the nice presentation. So I have a question. Can you go to slide number 12?
Yes. Can you see it?
Yeah, yes. So see, compared to your uncorrected image and your corrected image, the amount of aberration is not very significant. So I was wondering, see, you have these points, and these points in the uncorrected image, they appear as distorted.
Yes.
But these points are, symmetric in the corrected image. So can you use these, points, these aberrated points as PSF? And why not use deblurring techniques? There are different types of deblurring techniques.
So this has been a big discussion in our when we published this research. People have done this work where they use you can say the learning, like the PSF estimation techniques, which are basically that estimate that how does a point object will distort. But this doesn't happen all the way. The image that I'm showing you, which is basically a brain sample where the neurons are present and very tiny structures are present, yes, it will work. But when you go to a complicated structure, not complicated, the other structures, whether we are going to, suppose go to a lung structure, or suppose you go to a cochlear structure where hair cells are present, or in the lungs, you have the microvascules are present. There, these structures are not similar. So that's where the other techniques, which are trained on just very points, PSF estimation technique, or on the point object, they start to fail compared to our technique, which is being used. Yes, our technique is very new. We need to keep on training it for further, more samples, more structural variancy. But yes, that's what our technique differs it from these PSF estimation techniques.
Yes, thank you very much.
Dr. Vinod, you are muted.
Can you go to your slide number 11, experimental system?
Yes.
So you are basically collecting the aberration in the deformable mirror, right?
Yes.
So that so when you collect the aberration at the deformable mirror, so once you add the sample, then there might be a chance to treat that sample itself as aberration, right?
What do you mean by treat the sample as aberration?
Because as I said, because this setup is completely looks like new. What we read is like the illumination aberration correction is what is familiar. This is quite interesting, this kind of setup. So that's what I would like to know. If you collect the aberration at the place of a deformable mirror, so when you add the sample or the sample medium might also create another aberration, right?
Okay, I understood what you're trying to say. You're trying to say when you add a medium, how do you remove the baseline?
Yes.
So that's so the baseline is basically, it's done how is you add your imaging medium, which is basically in this a chemical compound, and you go to your surface of your sample. So at the surface, the assumption comes that the aberration will be minimalistic because the light is not traveling through your specimen. So at the surface, when you are imaging, just to give you an example, if you image at here, at this location, the light only has to travel through the medium. So there you estimate your baseline, and that is the part. It's like your like using, it's like a DC value that you give it to your deformal mirror.
Oh, now
And
I
from
got it. Now I understand.
from there you add
because
the
this
whole
point
thing.
you missed it. So yeah, I understood. So
Yeah.
you already have estimating the baseline.
Yeah.
That's excellent.
So that's the thing. Like the baseline will change from one microscopy setup to another. So that cannot be universally trained.
Yeah,
So that you need to remove it.
Smart adaptive light sheet microscopy. Yeah. It's impressive. Yeah, let's. If there is no further question, let's thank the speaker again.
Thank you, everyone.
Yeah, thank you, Dr. Mani.